For over a century, the medical understanding of Alzheimer's disease has rested on a straightforward sequence of events. Misfolded amyloid proteins accumulate, form dense physical plaques, and trigger a cascade that ultimately destroys a person's cognitive function. But when pathologists began looking closely at large populations of aging brains, like those in the landmark Katzman and Nunn studies, the physical evidence completely broke that sequence. They found brains overflowing with neocortical plaques that belonged to individuals who had remained cognitively sharp until the day they died. Up to a third of older adults pass away with enough amyloid burden to meet Alzheimer's criteria, yet show normal cognition. The amyloid wasn't cleared. The plaques were fully present, taking up physical space. They simply did nothing to the surrounding brain. This creates a biological contradiction. If amyloid is the definitive trigger for the disease, there must be an intervening mechanism that protects these specific, heavily burdened brains. Modern neuropathology provides an answer. Cognitive survival doesn't require a lack of amyloid. It requires amyloid that is structurally walled off from the rest of the brain. The execution of this defense comes down to a single cell type, the microglion. This resident immune cell acts as the brain's full-time gardener, and its capacity to maintain disciplined restraint is the exact variable separating a diseased mind from a resilient one. The determinant of cognitive survival rests entirely on the physical state of the immune system that meets the pathology. To understand how this immune defense works, we have to look at what is actually lost in dementia. The physical substrate of memory and cognition is the synaptic network. This graph illustrates the findings from Terry and Dukoski. It plots cognitive decline against physical markers in the brain. The scattered points show plaque count is a poor predictor of dementia. The tight correlation line reveals synaptic density is the true metric of cognitive decay. A brain keeps its mind for exactly as long as it keeps its synapses. So the question becomes how an inert deposit of amyloid translates into the active destruction of those connections. The damage originates at the boundary of the deposit. A raw, unstructured amyloid plaque has a diffuse edge. It continuously leaks a soluble, protofibrillar species of amyloid outward, creating a toxic halo that drifts into the surrounding healthy network. As this soluble halo touches nearby synapses, it triggers the complement system. It coats the healthy connections with a specific immune protein called C1q. This protein is a molecular kill switch. Microglia patrolling the area read these complement tags and aberrantly reawaken a developmental pruning program. They consume the tagged synapses, actively dismantling the mature circuits they were supposed to protect. The plaque itself does not erase the mind. The diffusible halo tricks the brain's own immune system into tearing down the hardware. This brings us back to the resilient brains, the individuals who carry the same heavy plaque loads but keep their cognition intact. Tissue analysis from these brains shows preserved synaptic markers and a remarkably quiet immune environment. Their microglia avoid the destructive pruning cycle by adopting a highly specific, targeted cellular program known as the disease-associated microglia, or DAM state. Entering this state requires a deliberate biological shift. The microglia must dial down their normal homeostatic patrolling behavior and pivot toward a protective, barrier-building phenotype. The result is structural. Microglia migrate to the nascent plaque, converge, and interlock. They build a continuous wall wrapping the amyloid. This barrier performs mechanical work. The interlocked cells compress the diffuse, leaking amyloid into a dense core, trapping the toxins inside. Outside that barrier, the environment remains clear. The surrounding neurites and synapses are completely insulated from the toxic halo, allowing them to function and survive unharmed. Cognitive resilience is this physical seawall. By containing the hazard rather than attempting the impossible task of clearing it, a disciplined immune system uncouples the presence of plaque from the onset of dementia. The ability to build and sustain this seawall for decades is governed by the innate immune genetics of the individual. When we map the complex genetic risk factors for Alzheimer's disease, they resolve into a straightforward grammar. Inherited variants predictably push the immune system toward either breaking this barrier or fortifying it. This diagram maps the microglial cell membrane, focusing on the TREM2 receptor. It detects amyloid and triggers the signaling cascade, shifting the cell into the barrier-building phenotype. But a mutation, TREM2R47H, causes a partial loss of function. The signal fails to fully engage. Without that signal, the microglial barrier remains sparse and weak. The plaques stay diffuse, the toxic halo leaks outward, and nearby axons suffer severe damage, roughly tripling the individual's risk for clinical Alzheimer's. Conversely, a mutation further down that exact signaling cascade, PLCG2P522R, operates as a gain-of-function variant. This hyperactive variant tunes the microglial cell heavily toward the disciplined state, increasing responsiveness, accelerating plaque compaction, and thickening the seawall. An individual's genetic risk for Alzheimer's is, in large measure, simply the genetic code dictating the structural integrity of their microglial containment system. The macrogeography of the brain introduces the second hallmark of the disease, neurofibrillary tau tangles. The origin of these tangles presents a stark reality. A hyperphosphorylated tau seed kindles in a small, poorly defended brainstem region called the locus ceruleus in nearly every human being by midlife. If this initial spark is nearly universal and the amyloid plaques are already present, we have to explain why the pathology stalls in the resilient brains instead of rapidly spreading across the cortex. This 3D connectome visualizes the progression of tau, known as Braak staging. This model of resilience directly explains the historical frustration of Alzheimer's clinical trials, particularly the long track record of failed non-steroidal anti-inflammatory drugs. Those broad immunosuppressive treatments operated on a flawed premise. They treated all microglial activation as a uniform negative, attempting to silence the brain's immune response completely. The physical consequence was disastrous. By indiscriminately stopping the immune cells from activating, the drugs dismantled the protective seawall along with the inflammation, releasing the toxic halo to freely wash over the synaptic network. Effective treatment requires phenocopying the fortunate by medically inducing the exact disciplined restraint seen in naturally resilient brains. This is the logic driving targeted therapies like TREM2 agonists. By engaging the signaling receptor early in the preclinical window, these molecules aim to lock the microglia into that active barrier-building state before the synapses are heavily damaged. This graphic maps the extreme sensitivity of that approach. The therapeutic target sits in a highly specific, narrow band of activation. It is a precise knife edge. Understimulating the cells leaves them dystrophic and insufficient, allowing the diffuse halo to spread. But overstimulating them pushes the microglia into chronic dysregulation, triggering the exact complement-spraying synaptic destruction the therapy is intended to prevent. The ultimate objective of Alzheimer's medicine is not to silence the brain's gardener, but to govern it. Survival depends on ensuring the immune system maintains the strict, disciplined restraint necessary to carry a heavy plaque burden without losing the mind.